Good afternoon, everyone. We're going to do microscopy today, and this is called a compound light microscope. Compound because there are two sets of lenses. Light because we have a light source down here, and microscope because we are going to use it to examine tissues on a very, very small level that we would not be able to see with the unaided eye. This portion of the microscope is called the arm, and this portion is called the base. And when we carry it, we use two hands, one on the arm, one on the base. It is a very, very heavy instrument, and so if you have any difficulty, ask your instructor for some help. The very first thing I want to do is to use the power cord plug this into an outlet. This is my on-off switch. I'm going to turn it on, and as I turn it on, you can see that lights will indicate the intensity and that we can see light coming out of our light source. This light is unfocused. So we have what's called a condenser. That is that right there. You can also see it right there. That is the condenser, and that is going to condense and focus light on whatever object I am viewing. To use this, I want to bring this, the stage, which you can see moving up and down, all the way down, and then I want to make sure that I have the shortest lens possible. And you hear the click? You can feel it also clicks into place. So we have lenses down here, and we have lenses up here. These are called the objective lenses, as they are going to be close to the object, and these are called the ocular lenses for my eyes, which is the ocular region of my body. So the very first thing I'm going to do is to look into this microscope, and you can see that these eyepieces or ocular lenses move back and forth. Nobody's eyes are the same width apart. So what I'm going to do is move those until they are right for my eyes, which means that I am going to see a single circle when I look through these. Now, I'm going to take a slide, and you can see it's a slide there. And I am going to put it on what is called the stage. And to do that, I am going to use my slide holder, this silver piece. This has a lever that goes back and forth. And it also has an area that is a 90-degree area where the slide will fit in. So I take my slide, I put it in and I let the lever hold it in place. Now, without even looking through the lenses, I am going to move my stage back and forth until I can see light going from the light to the condenser and focused on my slide where there is a slice of tissue. So I use these knobs to move the stage back and forth and left and right until I see that my slide is illuminated and that the slice of tissue is illuminated. The next thing I have to do is to focus my lens combination, and I will do that by raising or lowering the stage. Now you can see it rising rapidly. That's the outer knob. There's an inner knob. You can't even see it move. It's moving so imperceptibly. So these are the coarse focus and the fine focus knobs. So I'm going to bring this up until I just get it into focus, and then I will use fine focus. And I will make sure that I am seeing one single circle instead of two circles. That means both my eyes are focused. Now, we said this was a compound microscope. The ocular lenses are 10 power, and this short lens with the red stripe is 4 power, and that's called a scanning power. So this will bring my slide into focus, but it won't magnify it very much. The amount of magnification is the multiplication of the lens times the lens, the objective lens times the ocular lens. So I have 4 power and 10 power. So I'm looking at this at 40 times its normal size. And when I do that, I can make out that there's tissue, but I can't really see any cells. And of course, tissue is made up of cells. So the next thing I would do is click in the next lens, which is a little bit longer lens. And this is low power, and it is 10x, magnifying 10 times my oculars. 10 times 10 is 100. And I will again look through my ocular lenses, and I may have to adjust ever so slightly with the fine focus knob to get everything into focus, meaning visible, not blurry. This is not enough to see individual cells. I can see the nuclei of cells, so I know there are cells there, but it's not high enough magnification to see what I want to see. So I will again click into place the next longest lens. This has a blue circle, and it is labeled 40 times. 40 times 10 is 400 times magnification. I will again look through my oculars. It's almost in focus, so I'm going to use my fine focus. And now I can see individual cells. So I have magnified the slide, the tissue, again and again and again. 40, 100, 400 times. And at 400 times, I can see individual cells. But a couple of things have happened. As I've increased the magnification, I've decreased how much of the slide I can see. That's called field of view. And as you magnify things, you're going to see less and less of the slide. So I may have to utilize the stage knobs again to get back to my area of interest. Now, your microscope in one lens or the other will have a pointer, very much like my stylus here, my pointer, and you can actually move the stage back and forth until the pointer is on a particular cell. And of course, we've seen that in both lab and lecture when we did the histology portion, or you will see it when you do the histology portion. The other thing that happens is that since a tissue has depth, you may have to use fine focus to get the exact depth in the tissue where the cell that you're interested in resides. So I may have to look again, and using focus, bring out a particular cell. Now, when I do that, other cells are not in focus. Having done that, I may have to make an adjustment or two. Now, the other thing that's happening as I magnify, or as I increase the magnification, when I move the stage back and forth, either left or right or forward or backward, the stage is going to appear to move very, very rapidly at high power and very, very slowly at scanning power. Finally, we have another lens, that's this lens, which is banded in white and is 100 power times 10 is 1,000 times, but I can't use this lens without adding a drop of oil to my slide. This is called an oil immersion lens. We use it very, very rarely. We use it when we want to see tiny little cells like red blood cells, something of that nature. And to do that, I would bring my stage down, halfway move the lenses, put a drop of oil there, move the oil inversion lens until it clicked into place, and I will feel it and hear it. And then I would bring the stage up, and I would bring it up until the oil immersion lens touched the drop of oil. I could then look in my ocular lenses and just using fine focus, get the object of my interest in focus. Now, the condenser, that's this portion, should not be touching the slide. So I have a way of moving the condenser, and I'm just going to put a small lens. So we have a knob here that moves the condenser up and down. And I want it without a slide in place, up as far as it will go. And then I want to bring it down just a little bit so that when I put my slide on, the condenser is not going to touch the slide. The condenser also has, and let me show you this because you can see more light, less light. This has marked on it how much light you should use for each power. So as I go up in magnification, I need more light coming through the condenser. As I go down in magnification, I need less light going through the condenser. So this is just like your f-stop on a camera, which allows more or less light, but it's also going to make the slide, the slice, the tissue, more visible at a certain power. When I'm done, I want to do a couple of things. I want to make sure the small lens, the smallest lens, the red stripe lens, the scanning lens, the 4-power lens is in place. I want to move my stage down. Then I can take my slide off. I want to use my switch to turn the light off completely, and I will see the last light go out. I will see the light from my light source go out, and I will feel a click. My microscope is not yet ready to put back into place. I have to take the power source, wrap up the cord, stow the cord in the back of the microscope, and there is also a place where your power cord fits in. And now the microscope is ready to put away. So to put the microscope away, again, I'm going to use the arm and the base and use 2 hands to lift it and take it to the cabinet where I got it from. And the microscope will have a number on it, and the shelves of the case will have a number on it that matches the number of the microscope. And that concludes our talk on the microscope.